Carol, I think you're muted.
Hey, everyone. So excited that my friend Dr. Stempien-Otero has agreed to join us today from the University of Washington. I'm going to turn it now over to Ashley, who will introduce her, and then we'll get started. Thank you again, April, for joining us.
Thank you for inviting me.
So today for Grand Rounds, we've got Dr. Stempien-Otero, who's the Craig Tall Family Endowed Professor in Heart Failure Research and University of Washington Associate Professor of Medicine. She's a heart failure transplant cardiologist with extensive experience in both clinical and basic science research. She earned her bachelor's from Dartmouth and an MD from the University of Connecticut, and then medicine residency and chief year at University of Virginia, followed by University of Washington for cardiology fellowship. Her basic science research focus in the past has been the role of stem cells and cardiac repair and regeneration. She's received numerous awards, and today she'll be presenting Grand Rounds with a talk entitled Little Fires Everywhere, Inflammation and Chronic Heart Failure. So thank you so much for joining us today, and we look forward to your talk.
Thank you, Ashley. Let me share here, and let me go to this. Is it all working? Everyone can see my slides. So this is actually the t-shirt my son came to dinner with last night. He had no idea that I was doing this talk today, because of course he's an oblivious teenage boy, but he's super sporty. And so I think he acquired this from one of his friends. He had to point out this was the Russell Westbrook years. So this is vintage 2008. But yeah, I'm going to talk about immune activation and chronic heart failure. And this is a very broad talk. It's meant for more clinical teaching audience. So if there's some basic researchers in the audience on my favorite molecule, and I don't mention your favorite molecule, I apologize. It's meant to be kind of an overview of what's going on with inflammation and chronic heart failure talk. So this came out in the Atlantic during the COVID, and I just loved this when I saw it. Immunology is where intuition goes to die, because as soon as COVID occurred, I was like, and it started being obvious that it was affecting the heart, I'm like, oh my God, everyone's going to get it now. But it just unleashed for us how complicated the immune system is. And in this article was a great joke about immunology, in which an immunologist, cardiologists are kidnapped. They're who are we going to kill? And the cardiologist says, well, I've identified drugs that have saved the lives of millions of people. The kidnappers turn to the immunologist, say, well, what have you done? And the immunologist says, well, the immune system is very complicated, probably about to go off into a long explanation. So the cardiologist says, just shoot me now. And outside of my transplant colleagues, when I start talking about inflammation and immunity, that's sometimes what I feel like people's response is. But I just want to convince you that it's important for those of us that treat heart failure to have a working knowledge of the immune system and the heart, because it's really, to me, a next frontier for treatment of heart failure. So we're going to immunity, I missed this one. Role of innate immunity in homeostasis, acute injury and chronic disease, and then some mediators that we could potentially target in future directions. So I like to simplify the immune system into two compartments: the innate immune system, the adaptive immune system, and think about them as Mr. Clean and Marie Kondo. So the innate immune system is what immediately jumps into action and cleans and disinfects, spreads bleach over everything indiscriminately. The adaptive immune system is much, much pickier and more precise in its activities. So the innate immune system, very old program, goes all the way back to worms, shows its role is the rapid detection and containment of tissue injury and infection, and then a transition to tissue repair and regeneration very rapidly. The adaptive immune system did not appear until jawed vertebrates from a phylogenetic standpoint, and it's developed for antigen-specific response. So it has multi-pronged strategies, but it is specific to whatever antigen is being injuring the creature, and then adaptive immunity to have memory so that the next insult is treated faster. So what, vaccinations, right? The primary cell in innate immunity is the tissue macrophage. The trigger is, again, broad. It's just protein structures. It's not very specific antigens. So there's a variety of injury proteins and molecules that trigger an innate immune system. And again, it has no ability to detect self versus non-self. It just looks at proteins, motifs. Adaptive immunity is very trigger specific to very small epitopes or protein sequences, such as in transplant and HLA that's foreign to the body, for example. And this leads to its ability to differentiate self from non-self. The innate immune system is where I've done my studies because it's very simple. You have an injury, you have the risk of having an infection, like a bacterial infection. You have, in this case, this is skin, which is the simplest. So you have a tissue macrophage, which in the skin is a Langerhans cell. It's activated by the injury that occurs. It stimulates release of cytokines. Then that calls in more cells, granulocytes and monocytes into the area to both kill any bacteria immediately and heal the injury. The adaptive immune system, on the other hand, is quite a bit more complicated. And I'm not going to say that the adaptive immune system doesn't play an important role in cardiovascular disease and atherosclerosis and in heart failure, but we're just starting to understand it. And because of its complexity, I would put you all to sleep if you went on it. It really requires looking at just at very small parts of it to get a grip on it. What is common, though, I just want to point out between the two systems is the tissue macrophage. And so anything that I'm going to talk about with tissue macrophages and monocytes in innate immunity in the heart, you have to keep in the back of your mind that is clearly those tissue macrophages are likely affecting the adaptive immune system. And so that there are going to be increasingly papers you're going to see that are going to be based on adaptive immune responses in the heart that are based on the fact that this is the core cell. The macrophage has multiple roles in the innate immune system. It doesn't just do one thing. It goes in waves. There are predominantly two types of macrophages. This is an oversimplification. It's more like a color wheel. But the current understanding is that in response to an acute injury, you get inflammation with M1 macrophages. These are the macrophages that we all learned about in medical school, that they make tumor necrosis factor, INOS, very pro-inflammatory proteases, degrade the injured area, kill all the bacteria with INOS, and then set the stage for the next group of macrophages, which are the M2 macrophages that allow repair of the tissue. They elaborate a different set of cytokines, predominantly TGF-beta is the one would hear the most about, and they initiate, on top of cleaning up where the area has been cleaned up, laying down matrix and allowing in the sort of prototypic resolution of innate immunity that the injured tissue area is regenerated. So in the skin, for example, if you have an injury, you're going to have the M1s come in, they're going to clean everything up. You're going to have the M2s come in, they're going to signal to fibroblasts to lay down collagen so that your matrix gets laid. And then your regenerated skin is going to come into the area, fill in the area. You'll be left with a little bit of matrix in the scar, but then the entire injury stops and resolves so that you don't have continued inflammation. But this gets disrupted in situations where you have lots of injury all the time. So in chronic injuries, and I think an easy thing to think about from the skin perspective is chronic tissue stasis ulcers, where you have continued ischemia in an area, edema causing pressure in the area. You're going to have continued inflammatory injury stimuli, and then you're going to have continued stimulation of both inflammation and repair. In addition, in tissues that don't regenerate, you're also going to have issues because you can't have the signals to stop this. And in both these cases, you're going to end up with both signals, the inflammation signal and the repair signal, leading to chronic inflammation and fibrosis. And so it's also important from an overview perspective to know when we talk about a condition that causes chronic inflammation, that although the inflammation causes proteases and degradation of tissue, it's also at the same time frequently stimulating scar tissue development and fibrosis. So you get a little bit of a teeter-totter where these two things are happening at the same time, and you can get different rates of inflammation and fibrosis depending on the disease process, and then of course all of these different cytokines and mediators. But again, you go to like a chronic ulcer and you're gonna have a lot of inflammation, a little fibrosis around the edges, the thickening of the skin, et cetera. So that would be a good example of when the resolution is impaired and where you get immune dysregulation. And so again, if you think about the Mr. Clean with chronic inflammation, you get too much bleach in the area, you're just going to get irritation and constant inflammation and trying to clean it up, and it's just going to irritate the area and feed upon itself. Now, I'm just going to say flip side, the dysregulation in the adaptive immune system is Marie Kondo not being able to figure out what's self and non-self, and leads to autoimmune disorders. You know, she's just piling up tons of socks that she loves or doesn't love and can't decide because that pathway's been blocked. And so the dysregulation on that side leads to autoimmune disorders. So where does the heart come into this? Well, the heart's a lemon, right? It gets chronic injury and it can't regenerate very well. And so that's what leads to this role of inflammation and heart failure. So first, let's talk about homeostasis. So we don't think about the heart as having immune cells. Everyone always thinks about the muscle cells in it, and sometimes they're now thinking about the fibroblasts. But resident cardiac macrophages are fairly abundant in the healthy heart. And these are human tissues from a paper where you can see that they stained with GFP for macrophage markers. And you can see that macrophages are pretty common if you do the right stains and very carefully. They're very small, they're very thin, they're kind of, you know, spread out amongst the heart tissue here. And so it's easy in just like regular sections not to see them. And that's, I think, why they were ignored for many years. And they have multiple roles in homeostasis. So they're very important in tissue, in matrix turnover. So anytime, you know, collagen turns over about every 6 days in your heart. And so as collagen breaks down, it needs to be protease degraded in a controlled fashion, taken away so new collagen can take its place. Again, low-grade tissue repair. And then cells turn over in the heart too. So it's important for cell turnover and clearance to have resident macrophages to regulate all this. And I think if anybody wants to see a great example of this super paper from Cell in 2021 talks about the role of cardiac macrophages and cardiac homeostasis in mitochondrial mitophagy turnover. So this was a paper that was a mouse model, but it very elegantly showed how mitochondria normally age out, and then they are exocytosed out of the cells into the interstitium, and there they are recognized by resident macrophages to be eaten. And if you disrupt this system, you actually get too many. You have a buildup of old dead mitochondria that disrupts the system and causes heart failure in this case. But really good paper. And it actually comes with instead of a graphical abstract with just a picture, Cell now has little movies that they give. So I really highly recommend look at this paper and this little movie. And then here's another paper that shows another side of resident cardiac macrophages, and that's their response to stretch. So that as the heart stretches in a normal fashion, and this is just stretching out cardiac macrophages in tissue culture, that you get increased growth factor expression from them, so from cardiomyocytes. So IGF, HBEGF, note those are both two factors that we know are important in homeostasis and cardiomyocyte growth, because when they're blocked with chemotherapeutic agents, you get cardiomyopathies. And then if you block stress receptors here, these trip things, you block this response. But I think this is a great example of a resident macrophage that's just doing its job in the heart, keeping things happy. But what happens in injury? And as I talked about with the innate immune system, its classic role is in tissue injury. And a vast majority of the research has been done looking at injury, in particular myocardial infarction in the heart. And so I'm only going to spend a really small amount of time on this just to set up the chronic issues, because there's just myriad of papers I could go through, myriad of arguments in the field as to whether a total occlusion of the LAD of a mouse versus ischemia reperfusion injuries in mice are better. But this summary, I think this is the best summary from Doug Mann, who has shown in multiple different papers how tissue injury in the heart leads to the elaboration of these multiple factors that are all stimuli for the innate immune system. So some of these factors are pieces of things like a hyaluronan fragment. Some of these factors are things that normally live inside a cell and they're not normally in the extra tissue matrix, like double-stranded RNA, which should be inside a cell, inside a mitochondria, but in cases of injury is released. ATP even itself, as it's released from injured cells. All of these are stimuli of toll receptors, which are the primary receptor in the innate immune system, and then can activate the innate immune system to bring in more cells and do the Mr. Clean on the area. And I'm just giving this as a self-serving example of our own research in my lab, where we did myocardial infarctions, and we did 2 different manipulations of macrophages, specific to macrophages, where we made them more M2 or more pro-fibrotic, or less M1, so they didn't have that first stage of injury. And you can see here, if you don't have that first stage of injury, you actually have less healing, because then you can't call in the second stage than with a normal myocardial infarction. And then if you have too much of that second stage, you get too much fibrosis here. So this is just an example of how when you have these phases of activation of monocyte macrophages, you get different responses. And then within a given heart, one can imagine that as that regulation or misregulation goes on, you can get in some patients a ton of very thick scar tissue. In this case, this looked like it was a better ejection fraction after the myocardial infarction, but these mice also had more fibrosis and uninjured myocardium. These mice had less fibrosis overall, but that led to more dilation of their hearts. So there's clearly, even in acute injury, this balance between healing, resolution, is key in determining what's going to be the phenotype in the particular heart in the acute response. And you can imagine that that's just going to be accelerated in the chronic response, because loss of cardiomyocytes, as we know, is a major factor that leads to heart failure, and also is something that occurs continuously during heart failure progression. So let's go to heart failure progression and chronic heart failure. So as I said, you get loss of cardiomyocytes, which is going to promote the initiation of heart failure, promote the initiation of this innate immune response. It also occurs in response to other causes of heart failure. So I look at heart failure as being caused by loss of cardiomyocytes or increased wall stress, i.e. when you get chronic hypertension, chronic valvular disease. And increasingly, we understand now that most of the sarcomeric mutations that cause genetic cardiomyopathies also interfere with wall stress on the myocardium. Of course, loss of cardiomyocytes and loss of tissue in an area and scar development is going to increase wall stress over here. So independent of having hypertension, you're going to have increased wall stress with other cardiomyopathies. And these cardiomyopathies over here are going to lead to death of cardiomyocytes. So this essentially is giving you a setup for that chronic stress and chronic persistence and activation of macrophages within the heart, the macrophages that were there as the innate immune system, over time get replaced by bone marrow derived macrophages, which are even more pro-inflammatory. They lose those homeostatic ability to just control things and slowly turn over. And then they continuously turn on these M2 macrophages. Again, the heart, I don't know if there's any regenerative people in the audience. I know there's some across town from you. But the heart is essentially non-regenerative. And so you have a setup with chronic heart failure of simultaneous cardiomyocyte dysfunction, but simultaneous chronic inflammation and fibrosis. So this all makes sense now, but we've actually known this for a very long time. So these are the initial studies that showed increased inflammation in chronic heart failure. The Levine study from the New England Journal, which looked at just all comers with congestive heart failure, looking at tumor necrosis factor levels, and then other studies looking by New York Heart Association at tumor necrosis factors levels. And this has also been repeated with several other pro-inflammatory cytokines over the years. And I think it's important in looking at the history of this to know that these studies were initially felt to be due to cardiomyocytes elaborating TNF, which they do, but the role of the immune system in the heart at this time was a little bit poo-pooed and not felt to play a role. And so I think it's been an interesting story over 20, 30 years to watch how the realization that there are different cells in the heart other than contractile cells and their contribution to acute disease. There's also a long bit of supportive epidemiologic data to this, and I've just picked out some of the hundreds of data just because I think that it's the most pertinent to clinicians. One is what I mentioned, that you have a drop off in these resident macrophages over time. They're replaced with these inflammatory bone marrow derived macrophages. Corey Levine, they showed in patients with worse heart failure and human cardiac tissues that you had this gradual loss of these particular resident cells over time. We know that other conditions that perpetuate inflammation in the body also make heart failure worse. So patients with risk factors for systemic inflammation, like diabetes in particular, have worse outcomes with heart failure. The CANTOS trial, which was the IL-1 inhibitor, had a suggestion that inhibiting inflammation could attenuate heart failure development. The big problem is that drug was so expensive that really the appetite for additional trials was quelched. And then we know that specific immune responses to stress may direct this transition to a more stiff heart versus a dilated heart. And I'm going to go into that a little bit more later. Again, heart failure, I just want you all to think of it as it's a chronic inflammatory state. And when I started doing my crazy stem cell studies, injecting bone marrow into hearts, it became apparent to me, looking at tons of slides, just how many macrophages in regular advanced heart failure tissue there are. And that's really what got me interested in looking to them more specifically. And you can see here, this is these deep brown spots are the macrophages. These big things are cardiomyocytes with their nice big nuclei in them. And you can just envision that these macrophages are just continuously crawling out of the blood and setting up home within the myocardium, causing havoc. Again, as I mentioned, these populations change over time. Corey Levine, if anybody wants to do sort of a deep dive, he's got the best research on this. This time it was a protege of Doug Mann. They just published in a new journal. There's a new journal out there, everyone, Nature Cardiovascular Research, first issue this year. But this looked at single cell RNA-seq on isolated cardiac macrophages from hearts. Well, single cell on whole heart, in which they looked at the macrophage subset defined by macrophage genes being expressed. They looked at donor hearts, normal, being used for transplant. They looked at dilated cardiomyopathy hearts. And you can see here how the genes expressed by the macrophages change quite a bit between the resident state and the disease state. I think this shows best this overlap in color. There's actually very little overlap in color. In addition, if you look at this, these are proliferating macrophages. In the normal hearts, there is quite a lot of proliferating self-renewal of these resident macrophages, whereas in dilated cardiomyopathy, this fraction decreases over time, as shown in these other studies. So this is clearly an important part, is not just what is being expressed, but the type of cell that's coming in and expressing it. So in conclusion, in heart failure, what we have is this chronic inflammatory state where you have release into the system of these factors that go into the circulation as cells die. They stimulate different monocyte populations to go into the cardiac tissue. Some of them degrade myocardium, degrade ECM and myocardium to some extent. Some of them are pro-fibrotic, and you get a balance overactivity of these back and forth, which is going to continue to promote worsened heart failure progression. Obviously, if you have more fibrosis, you're going to have more wall stress, which is going to perpetuate this. If you have more degradation, you're going to get dilation, which is going to increase wall stress in another way, and again, perpetuate this process. And again, the problem in the heart, because of its activity as an organ that we all know, it's continuously under wall stress. It requires huge amounts of mitochondria for function. So any disruption in that's going to cause lots of redox changes. You're going to have death without replacement of cells. And then in patients with ischemic cardiac disease, you obviously have the extra factor of patients having chronic ischemia, hypoxia, that's going to perpetuate this. So in summary, in innate immunity, we know that cardiac macrophages have a homeostatic resident role, but they transition in patients who have cardiac disease to worsen this chronic inflammatory stress. The myocardial stressors speed this process, and then you have recurrent release of pro-inflammatory factors that precipitate the state. So what do we do? It sounds grim. As I'm saying this, I'm like, oh my gosh, this is like almost as bad as several patients we have in the hospital right now, where we're just like looking at each other going, what are we going to do? Early on, we discovered that the immune system is not the same as the neurohormone system. And so at first, when the studies showed high levels of TNF, just like high levels of angiotensin, everybody thought, well, we'll just block it and everything will be better. And that didn't work. So these were the two trials that were set up around, let's call it the turn of the century. When we were fellows, Arnold Bass, the Recover and Renaissance studies that used the TNF inhibitor that was the first one that came out for rheumatoid arthritis in advanced heart failure. And as you can see here from the Kaplan-Meier curves, there was actually absolutely no benefit whatsoever. And with time, some of the other TNF inhibitors that were used for other diseases like Crohn's disease actually had worse heart failure outcomes in patients who happen to have heart failure. So this was a real wake-up call that we couldn't look at the immune system as just block one factor and it gets all better. But what we have learned over time is that it is fungible. We can change the macrophages within the heart. And that's by looking at other treatments that we're doing. So this was a study that my lab published several years ago, where we actually were focusing on fibroblasts with LV unloading to show that fibroblasts would stop making or change collagen production, looking at fibrosis with LV unloading. But what we found when we looked at the macrophage fractions is that actually the macrophage phenotype changed dramatically after unloading. So you can see here, the blue are patients that had primary transplant or LV cores that we collected and isolated macrophages from, and then the red were bridge to transplant patients where we did the same thing. So tissue was taken and we isolated the macrophages and did the gene expression. And these are not matched samples. So these are not necessarily, oh no, they are matched samples. and 8 to 16 pre and post. No, take it back. Somebody's going to get me. Some of the scientists is going to get me. Not match samples, otherwise we would have the same number, right? So these are a group of idiopathic patients that had primary transplant or VAD, and a group of idiopathic patients that were bridged to transplant unloaded. So macrophages from loaded tissue and unloaded tissue, and showed that we had a significant decrease in expression of these pro-fibrotic factors in those macrophages. We had a drop in one specific protease and a trend towards a little bit of increased inflammation. Although this was not statistically significant. Looking at pro-inflammatory factors is challenging in human populations because there is genetically a wide variance. But it was clear that there was changes in macrophage phenotype from unloaded hearts in comparison to loaded hearts. And our conclusion from that is that although there was no change in fibrosis, that there were ways that you could change macrophage phenotype within the diseased heart that should be addressed. And intriguingly, similar studies done in mouse models showed also that there were these changes in macrophage. This was looking at a sarcomeric mutation model. So this is a human sarcomeric mutation in a mouse. And they looked at this TRP channel, which is a stretch channel. And when they inhibited the stretch channel in macrophages, they were able to attenuate remodeling. So changing the macrophage response to stress, similar to what happens with unloading in the LVADs, change remodeling. And I know that this group is kind of going on a whole interesting deep dive on whether or not you can specifically modulate these stretch receptors and change macrophage phenotypes within the heart. I think it's a great avenue to pursue. Finally, I want to talk about another way that nature changes macrophages in humans, and that is sex. So COVID really unwrapped for us the difference in immune response between men and women. As you can see, these are from the first wave. This is early data, but it's continued that men have much more fatal disease from COVID in comparison to women. And there are likely confounders of maybe men aren't going to the hospital sooner, et cetera, but it's been really persistent and consistent data. The flip side is that the incidence of long COVID appears to be much more in women versus men. And this has been known for a long time that there's a difference in immune response between men and women. This is from Nature Reviews Immunology in about 2016. And you can see that there are definitely biases in diseases that are known to be immune mediated between men and women, in that women tend to have more autoimmune diseases, and men tend to have these non-reproductive cancers, which are thought in part to be lack of immune surveillance, blocking these cancers from developing. What's super interesting here under infectious disease is that MERS, which was the Mediterranean coronavirus, already known to be a male bias at this point. And this was obviously way before COVID came. So we know that these diseases are female predominant, and these are male predominant, and it is really due to differences in the immune response between men and women. And the corollary of this, and this was in this very new paper, and I was really gratified to see this because it's been a hypothesis of mine. My favorite thing is when people test my hypotheses and I don't have to spend my own research money on it. And this is looking at primary component analysis. So this is single cell RNA genetic analysis of macrophages from that other study I showed you from Levine lab. These are women, female donors with normal hearts. These are male donors, normal hearts, dilated cardiomyopathy males, dilated cardiomyopathy females. Again, this is all macrophage cells. And when we talk about principal component analysis, it's really how much certain genes are expressed and where does it fit in a component analysis. And you can see that they separate out not just by the dilated cardiomyopathy genes tend to fall over here, and the normal heart genes tend to fall over here, but quite strikingly between men and women, which I believe supports this hypothesis that men and women's immune systems function differently, not just in these diseases like rheumatoid arthritis, but could function differently within the heart and within heart failure and remodeling, and may explain some of the differences in the incidence of HFpEF and HFrEF between the two, and outcomes between the two sexes. Finally, I just want to throw in another external factor that mediates the immune response, and that's social determinants of health. It is increasingly understood that various stresses, social stresses such as racism, poor socioeconomic status, poor diet, all lead to increased norepinephrine signaling within the central nervous system, which also, as a downstream, increases pro-inflammatory and decreases anti-inflammatory genes within the body. And so this is another avenue I'm not personally researching, but I think it's an avenue that research could help uncover why our patients who are under social determinant of disease stress may have worse outcomes with heart failure. So what are our future directions? So, there's plenty of mice research going on. I think there's a real limitation in heart failure research in mice in that, especially with regard to inflammation, is that there are limitations in similarity to the human immune system. I mean, essentially mice live in the dirt, and so their responses are very different to ours. This was shown in about 10 years ago in a study where they did cecal punctures and showed that mice's immune response was very different than the human response to things. That you can have humanized immune systems that help with this. I think there's also the challenge that models of chronic heart failure, which is what we clinically deal with, are hard to develop in a mouse who only lives for 2 years. In humans, you have the problem of genetic variation. I've just shown you this big variation between men and women. There's also equivalent variation from person to person genetically, and developing appropriate controls it can be a challenge. We're looking at, if you compare normal hearts to somebody with heart failure for 20 years, so much difference between has gone on that you're really hard to understand the step A to B to C to D to F to Z mechanism of the diseased heart versus the normal heart. So I think that we need to focus on exploiting differences like this sex difference I talked about, and focus on taking patients with heart failure and studying them mechanistically, like with unloading studies. So here's an example of 2 research projects that we're involved in here at University of Washington. The first is looking at reducing systemic inflammation by improving mitochondrial function. I talked about how mitochondrial dysfunction leads to inflammation. My collaborator Rong Chan's lab has shown that nicotinamide riboside, which is a NAD donor, actually increases NAD stores within cells and then feeds their mitochondria, and they have better mitochondrial function. And so what we are doing right now is taking patients who are scheduled for an LVAD and 5 days before start feeding them either placebo or nicotinamide. And then we are harvesting peripheral blood and cardiac macrophages at the end of this. And also we have many other endpoints, but my particular aim in this project is looking at this comparison and looking at whether or not if we make the cardiac macrophage mitochondria happier, so to speak, with the NR, are we going to improve and change their inflammatory profile? In the preliminary data that Dennis Wang, one of our cardiology fellows, published in Journal of Clinical Investigation on the peripheral blood, showed actually a dramatic decrease in the inflammatory response of peripheral blood cells in response to inflammation. So we know that NR supplementation decreases peripheral blood inflammation. Our next step is just continuing to enroll patients to collect enough tissue cardiac macrophages to see if it's going to decrease inflammation within the heart. And then, hopefully can we use peripheral blood markers as a surrogate for what's going on in the heart. A second study that we're just starting is looking at Tavr patients. To me, this is a better unloading model in some ways than LVAD because the patients are less sick and their unloading occurs immediately, right, after the Tavr, and they're not hospitalized or anything. So you have a time point at baseline, you do a Tavr, and the next day they're going to have a change in their unloading. And so we're collecting blood at baseline 1 day, 30 days, 1 year, and comparing with clinical parameters to find the extremes of response versus non-response and see if it correlates with different gene expression in the circulating monocytes. The great thing about this study is that 50% of the participants, because 50% of people who get Tavr are women, so we'll able to also do a men versus women comparison to maybe see if we can use this as a discovery mechanism to figure out the differences between the two. So in conclusion, I hope I've all convinced you that the immune system is super fun and easy to understand. It is just Mr. Clean and Marie Kondo, that the innate immune system is what plays a critical response in myocardial injury acutely, but that this kind of gets hijacked in chronic heart failure, and that resident interactions may be critical mediators of heart failure progression. And we don't have a magic bullet, but I think that this is a really kind of next frontier that we could develop new therapies for heart failure. And that's it. I know that was,
Wow.
and like I said, it's an overview. I'm trying to give you guys a big picture of where we're going with this.
That was fantastic. Thank you so much for that discussion. I especially love the Mr. Clean Marie Kondo analogy. I will be using that in the future. Do you have any thoughts about inflammatory responses by different racial ethnic groups? I know in African Americans, there tends to be a more autoimmune inflammatory disease. And have you thought about that in regards to heart failure?
Yeah, I think it goes back to that, my thinking is it's that social determinants of disease data that increased societal stressors have central nervous system effects that then increase stressors increase inflammation totally in your body. And that it comes down to more of a, in nature nurture. It's not really a genetic issue in races, but it's a social treatment issue in races that leads to those differences in diseases. It's really a very hard area to research, obviously, because of all the confounders, but I really support people who are doing that hard research right now. I think it's really important.
Great. Yes. Thank you so much. All right. Any other questions?
This is Alberta. I have a question. You really made a great point about understanding that immunomodulation and neuromodulation are different. But what evolved, as you know, in this early work with Doug Mann back in the 90s was understanding that neuromodulation seemed to give us good outcomes. When, we looked at, I used to do work on beta blockers, and we saw that there was less interstitial fibrosis. There must be an interaction of, does the neuromodulation somehow decrease the inflammation? Is there a relationship that we see as these therapies that have become our GDMT, ACE intervention, norepinephrine, aldosterone? How does this interact with the immunomodulation? Do we see differences?
So I think the most straightforward is the angiotensin system. It's pretty clear that angiotensin is pro-inflammatory. There are angiotensin receptors on macrophages and monocytes. And in animal models where you infuse angiotensin in like little mice, one of the earliest things that happens in the hearts is you get perivascular macrophage accumulation. So that is one that's a very clear-cut, driver of inflammation at the neurohormonal level. It's it's always, again, from the correlation level, it's really hard, because if you put somebody on beta blocker GDMT and their heart failure gets better, then also the whole inflammatory milieu chills out, and that gets better too. I think what's going to be interesting is ways to try and do the reverse. If you improve the inflammatory, would you also improve the neurohormonal, to figure out if those are two independent pathways or they're always going to be interrelated.
Great, thank you.
Any other questions?
I flame them and talk too fast.
No, it's just so much to think about. Well, if there are no other questions, I just would like to say another huge thanks for you joining us today. This is fascinating work and really phenomenal talk. Thank you for your insights and again, that fantastic analogy that I will be using again. All right, everybody.
Well, thank you. I'll just say, if anybody has any further questions, my email is super easy up here. It's just april@uw.edu. So any other questions, feel free to contact me.
Great. Well, thank you again. That was just a fantastic talk, and I can see all these accolades coming in the chat. So thank you for joining us, and everyone have a great afternoon.
Thank you.